Hello, I'm Brian Bothner, professor at Montana State University in the Department of Chemistry and Biochemistry. So we are looking at biology as a system versus as individual parts. And so this research takes us from the atomic scale of protein models and virus structure, which you can see here, to the living systems corresponding in cells of metabolites, proteins, and nucleic acids that really come together to make a functioning biological system. And so we use a number of biophysical techniques and analytical tools in our research. One tool that we use is mass spectrometry. We make great use of mass spectrometry in identification of metabolites, in understanding protein structure and function analysis, and also, importantly, in how systems integrate and how biomolecules communicate with each other. And so some of our research projects are involved with studying viruses such as hepatitis B virus and understanding the functionality of antiviral agents. We are also interested in studying bacteria and how they respond to antibiotics. The Bothner Lab is also part of the Biological Electron Transfer and Catalysis Energy Frontier Research Center here at Montana State University. In this research, we're focused on understanding really at the molecular level how electron transport works through protein, and specifically how the process of electron bifurcation works. And so with these studies, we're actually conducting biophysical analysis of proteins such as nitrogenases and hydrogenases, and we're characterizing what are the molecular switches that are used in electron transport and electron gating. So in our lab, two major areas of research are proteomics and metabolomics. And through these studies, what we're trying to accomplish essentially is understanding how cellular systems work by integrating data from different levels of biomacromolecules. So looking at metabolites and understanding how metabolites relate to proteins and how the communication between proteins and metabolites are actually changing the physiological processes that are occurring in cells.
Hi, I'm Katie, and I'm a first-year graduate student at Montana State University in chemistry and biochemistry. I am currently now accepted into the Bothner Lab group, and I will be doing research utilizing metabolomics and mass spec and how those things work together. And I am also a TA. I'm a teaching assistant for general chemistry here at MSU, so I'm teaching labs and also taking classes and then learning the ins and outs of mass spec and metabolomics.
Hi, my name is Luke Berry. I am a graduate student here at Montana State University in Bozeman, Montana. I am a PhD student working in the lab with Professor Brian Bothner, and the research I do primarily focuses on looking at the biophysical nature of proteins in different conditions. The way we look at this is by using an instrument called a mass spectrometer, and I use a technique called hydrogen deuterium exchange, which I then couple to the mass spectrometer, which then allows us to look at the structure and dynamics of the protein in different conditions. And this allows us to tell about ligand binding sites, mechanism, and all sorts of other information that let us really understand the structure-function relationship of different proteins.
Hi, my name is Rachel Rawl, and I am a third-year PhD student in the lab of Dr. Brian Bothner in the Chemistry and Biochemistry department. My research focuses on taking a systems biology approach to answer a biological question. And what I'm interested in is looking at how arsenic affects the bacteria that live inside our gastrointestinal tract. So arsenic is something that's relatively common as a contaminant in groundwater. And so there are a lot of people all over the world that are exposed to high levels of arsenic. So what I do is I look at the genes and the metabolites that are upregulated when certain types of bacteria live inside our GI tract. And so how I do this is I use mass spectrometry here in the facility at the biochemistry department, And I look at all these metabolites that are upregulated in response to arsenic. And what I do with those is I take those and I compare them to the genes that are also upregulated or downregulated. And I look at what pathways or what metabolisms are being turned on and turned off that can affect the host health. And I've had a really great opportunity here to learn the entire process of metabolomics here in the chemistry department. So I start at the beginning where I learn how to prep samples and I run them, and then I do the analysis portion as well. So I've learned how to do the entire thing, which has been really beneficial for me. I'm also appointed as a graduate teaching assistant, so I teach a lab twice a week here, and it's one of my favorite things. It's been a really good experience to learn material and then teach it to students and be able to mentor and guide them.